But it's the West Virginia time at work here. It's just never advanced very far. With his BS degree in agronomy, with a specialization in epidemiology, he then went on to Iowa State. That one I can say, in Ames, and he got a master's degree in plant pathology there, with again a specialization in epidemiology. And then I can say the next one real well, University of Nebraska. We got an applause on University of Nebraska. Okay, good. All right. With a PhD in agronomy, specialization again in plant pathology, and also a minor in statistics as well at Nebraska. He's held two positions since then. One is a postdoc at Nebraska, and then he also worked with a private company, Spray Tech Fertilization. I guess the best way to characterize him is through his career has been so far he's an epidemiologist. He, one of the reasons we are very interested in his background, and while we think he would bring strengths to Uvalde, is the broad variety of pathogens he's worked on. Starting off, curiously enough, with lethal yellowing of palms, Kevin. Right, exactly. We struggle with similar diseases here right now in South Texas. Worked on Colotoxicum on strawberries, a number of other things. Soybean rust, a lot of epidemiological studies on soybean rust. And then most recently, I guess, with the soybean cyst nematode. He has a strong background in nematology. And he's also worked on diagnostics of some of these diseases, various aspects of them. Again, I'm sure he's going to cover some of this. He also has some teaching experience. He's worked at Des Moines Area Community College, taught some classes, and so he really brings a lot of communication skills, I think. So with no further introduction, Dr. Perez Hernandez, we'll let you take it over.
Thank you, Dr. Apple. Thanks for the introduction. It's a pleasure to be here with you this morning, and I thank everybody for attending this presentation. I want you to think about what I'm going to read for 1 minute. So, microscopic roundworms-like organisms burrowing through the cells of plants, injecting in the plant foreign compounds through a hypodermic needle-like structure. Worms that penetrate the root and grow so large that are too large to be contained in the plant cell, and they erupt from the tissue. You know, reading these lines, it sounds like a plot of a fiction movie, but believe it or not, this is the basic biology of a pathogen that is the most economically important pathogen of soybean, not only in the United States, but in the world. So, just to give you a little background about the pathosystem. First, the host soybean is a top cash value in the United States. In 2014, it was grown in about 85 million acres. So, just to give you an idea, in Texas, cotton is a big crop, probably close to 10 millions. So soybean is grown like 10 times in a larger acreage than cotton. So the total crop value was estimated at 42 billion. The soybean cyst nematode was first found in the United States, in North Carolina, in 1954. And since then, the pathogen has spread to all the major soybean growing areas of the country. In Nebraska, it occurs basically in every county where soybean is grown. And for the past decade, last 2 decades or so, this pathogen is considered as the most economically important problem in soybean. An estimation of yield losses from 2003 to 2005 indicates that the economic losses were close to $500 million dollars in the U.S. In Nebraska, in 2014, it causes $40 million economic losses. This graph shows the top 3 soybean pathogens in the U.S. And as you can tell, SCN is by far the pathogen that causes the highest yield reduction. This SCS is a Fusarium virguliforme, the sudden death syndrome of soybean. So, soybean cyst nematode is a plant parasitic animal and an obligate plant parasite. So, nearly all nematodes, they live in the soil. They spend a portion of their life in the soil. But unlike many species that only inhabit the surface of plants, or some species that penetrate the plants and move, the soybean cyst nematode, once it gets into the root, it establishes a permanent feeding site and completes the life cycle inside the root. So it's considered a sedentary endoparasite. And it's a cyst-forming species which is very unique among nematode genera. And this is the ability of the female cuticle to transform into a tough sac-like structure that protects the eggs that are developed within the body of the female and confers the nematode the ability to persist in the field for longer periods of time, even when there is not a suitable host available. So the life cycle of this pathogen is relatively simple and consists of eggs, juveniles, and adults. Cysts that remain in the soil embedded in the soil profile for long periods of time. These cysts often contain from 40 to 600 eggs of the nematode. So each of these eggs contains an infectious juvenile of the nematode. So stimulated by substances released by the root, or sometimes by temperature, the juveniles break through the egg shelf and then they emerge from the root, from the cyst. So as soon as an infectious juvenile has reached the root, then the juvenile uses the stylet to penetrate into the outer layer of the root, and then it migrates towards the vascular tissue where water and nutrients are concentrated. There, the nematode picks or selects an initial first cell for feeding, and out of the opening of the stylets, the nematode releases a variety of secretions. These secretions affect the vacuoles, but among other organelles, it affects the nucleus. So it transforms the cells biochemically and genetically, and so the cell wall of adjacent cells dissolves, and a big joint cytoplasm is formed, and this is called a syncytium. So the nematode undergoes two molts, and when it comes to juvenile for the nematode starts a process of swelling. So at some point, some nematodes that are going to become males, they regain the vermiform shape, and they get out, they abandon the old cuticle and abandon the root and no longer feed. But in contrast, nematodes that become females, they remain attached to a source of nourishment, and they continue to grow and to swell until they are too big to be contained in the root, and they erupt from the root with the neck and the head being embedded in the tissue. So after fertilization, in the course of a few weeks, the female produces eggs, and the nematode has completed the cycle. So under optimum conditions of soil, of temperature and moisture, the length from infected juveniles to viable eggs lasts from 21 to 22 days. And the nematode can complete up to 4 generations per growing season, theoretically up to six generations. The symptoms caused by the soybean cyst nematode in heavily infested soils is normally stunting and chlorosis. But more frequently, in the field, this is the situation. Plants, it's very difficult to distinguish an infestation. So this picture down here is a side-by-side soybean resistant and susceptible variety growing in a heavily infested soil. As you can tell, it's hard to tell which one is which just by looking at the picture. So in this trial, the resistant variety yielded over 30% more than the susceptible. So the soybean cyst nematode was found in the United States in 1954. 60 years have passed, and research has been done on the biology, on the ecology of the pathogen. But what remains, but management of this pathogen depends almost exclusively in the use of rotation with non-host crops mainly corn, wheat, and oats, and use of soybean-resistant varieties. These two practices are the most effective strategy to deal with this pathogen. So from the molecular era, 1984 to date, vigorous amount of research on trying to characterize the parasitism genes of this nematode, has been done. But these two practices just remain as the main method to deal with SEN. So the rest of my talk focus on these two practices through two studies. The first study is an evaluation of the relationship of soil texture with the population densities of the nematode in corn rotation. And the second part is a modeling approach using a multifactorial perspective, not only texture but other factors. And study 2 deals with SEN-resistant varieties, and it's a very simple analysis on a spatial pattern of this nematode, the spatial variability in experimental settings used to evaluate varieties in the U.S. So the first part of the study dealing with non-host crop rotation. Corn is the main non-host rotation crop used in Nebraska. Mainly in is used mainly in annual rotation. Normally, when the populations of soybean cyst nematode are going to be assessed, in soybean, samples are collected at planting and harvest. So the population densities are evaluated at the beginning and at the end of the growing season. So there is an evaluation of initial population and final population. But during corn rotation, producers, they are more interested they are not particularly interested in what happened between the beginning of the corn season and the end. They want to know what happened before they plant the non-rotation crop and what happened before they're going to plant soybean. So that involves a growing season and an overwintering period. So many factors have been associated with soybean cyst nematode, and in general, soil pH, soil temperature, and moisture, they have been related to SEN, but most of the studies have been in controlled conditions, but how these factors interact and how these factors contribute to the consistent observed population reduction in corn is unknown. So the part 1 of the study focused on texture for 3 main reasons. Texture governs several hydrological, chemical, and biological properties in the soil. And it has been associated with SEN, with the soybean cyst nematode, but this association has been mainly qualitatively. But the relationship of specific proportions of sand, silt, and clay with SEN in corn rotation is not clearly defined. So our first objective is to characterize this relationship. Commercial production fields in annual soybean corn rotation and with a minimum population of 250 eggs per 100 cubic centimeters of soil were selected in the major soybean growing areas of Nebraska. In each field, 10 sampling grids were collected in a zigzag pattern systematically, and from each grid, 20 soil cores were retrieved to obtain a composite. Each of these sampling grids were georeferenced for site-specific resampling. Each field was sampled in May before corn rotation and in May of the following year after corn rotation. So the populations measured here are defined as initial population and their final population. I'm emphasizing that because I will be using the term initial population and final population in my next slides. Working with quantifying the populations of SEN is relatively simple and consists of a standard sieving techniques. The method is pretty standard. Cysts, they have the property of floating in water, so soil suspension is formed. The soil is crushed and agitated, so cysts tend to float, and through a process of decantation with a set of sieves, the cysts are retained. Then this cyst into another set of sieves, are crushed, and then the eggs are released and extracted and counted. So it's a standard. The egg counts, SEN egg count, is a standard measurement for SEN population densities. So in this case, an average population density was obtained per field. And another portion of the soil was used for soil analysis of pH, organic matter, salts, and texture. So for this particular part of the experiment, we end up with a data set having 5 variables. We talk about texture, but texture is the integration of 3 constituents: sand, silt, and clay. So we end up with this data set, and because we want to understand relationship in this multivariate data set, we use these 2 methodologies. And just let me tell you very briefly why these methodologies are about. Principal component analysis is a mathematical procedure that has the purpose of reducing the complexity of a data set into a more manageable, understandable data set. So it converts a set of uncorrelated variables into a set of related variables into a set of uncorrelated variables called principal components. It's used quite a bit in high-dimensional image analysis and also in face recognition. Cluster analysis is a methodology to identify objects that resemble in a population in such a way that objects within the same group are more similar to each other than objects within other groups. So in this case, we want to know, with these 5 variables, we want to know, and because the cluster analysis uses an algorithm that measures the Euclidean distance, in this case, between these variables, the analysis itself will foreshadow some information whether or not a population decline occurs in sandy versus silt or clay soils. So all statistical methodologies have a purpose and a procedure. And the use of principal component in our study was mainly to assess the multivariate assumptions and to verify the clustering. That was our main purpose. And cluster analysis, as I said, to identify groups of similar, exhibiting similar population declines. Normally, cluster analysis, it's common in cluster analysis to present a hierarchical tree diagram or a dendrogram. But because the process is exploratory in nature, it's better to verify that the data actually sustain that clustering. And we use simulation with 1,000 bootstraps using the method proposed by 2 Japanese researchers. So in these both methodologies, what is more important is how many and what they mean. So whenever we see a principal component and a cluster analysis, the main question is how many components are in this data set and what those components mean. How many clusters and what those clusters mean. So we started from a data set of 5 variables, and the analysis gives 5 components, but how many of those components are giving me some information about this data set? And we use a method called a script plot. In this case, it indicates that in this data set there are 2 principal components. But now what? I have my 2 principal components. What does that mean? The interpretation of the principal component is based on the eigenvectors, correlation coefficients, and p-values. And in multivariate statistics, eigenvalues and eigenvectors are discussed every day or all the time. But an eigenvector, we learned that in physics. A vector we see a vector like an arrow pointing in a given direction. So that has a length. So we can think of an eigenvalue as the length of the vector and the eigenvector as the direction. So if I have a vector going that direction, I have a vector going in this direction, and I force this vector and pull it up, so I'm changing the eigenvector of the eigenvector. But the eigenvalue, the length of this remains unchanged. So just to give you an idea, because the principal component analysis uses all the data set and looks the data in a different dimension. So in this case, based on these eigenvectors and the correlations of the variable with the eigenvectors, our interpretation is that that sand is more related to SEM population densities than silt and clay. And this has been a claim, mostly an observational, and this continues to be observational, but observational based on some more more formal quantitative methods. And the interpretation of the principal component too is that it represents SEM populations, and it shows that it suggests that SEM populations increase as sand decreases. So mathematically, a principal component is a contrast of the variables. Like in this case, this coefficient is the eigenvector of the principal component 1 with sand. The second coefficient is the eigenvector of silt, and so on. So in this data set, there were two clusters, and that was confirmed with at a 95% confidence levels. So one cluster with sand predominant soil, and another cluster with sand and clay predominant soil. So when these clusters are compared, we confirm that the SEM population, the initial populations, those populations coming out of soybeans are significantly higher than populations coming from silt and clay, higher in sand than in silt and clay. But the population reduction was not significant. So this conclusion, we hypothesized that in Nebraska, in half of the state, sandy soils predominate, and sandy soils, they have more aeration. So during if they have moisture, these soils freeze faster, and that probably causes mortality. But based on this, whether or not sand exacerbates or not mortality of SEN compared with silt and clay is unclear. So, and these are the specific ranges based on the data of why SEM populations occur in sand compared with silt and clay. So the part of the second part of the study deals with a modeling of SEM population density decline. And why to use models when we use models every day, but why to use a mathematical or statistical model to study a system? And the thing is that models offer a very robust approach to identify and understand relationships, and most of the time they have the advantage of elucidating complex relationships that are not easily distinguished with other methods. And eventually models offer the capability of predicting tools. So just to reiterate, there are many factors that have been associated with SEN in general, but and it's well established that during rotation populations decline, but the factors that are associated with such a decline are not clear. That's not clearly defined. So we use a modeling approach to identify what factors are favoring that decline during annual corn rotation. So the model was built in 4 steps. And for the first step, we already know that the final population is a response variable. And we have a set of 8 explanatory variables, including the soil analysis and tillage irrigation. Texture was converted into a texture index. And this is simple, but normally if texture is used as a explanatory variable in a model, according to our principal component analysis, and sand, silt, and clay are used at the same time, it can cause some problems in modeling, something called collinearity. And so collinearity causes computational problems and sometimes overfitting of a model. So meaning that some coefficients, some variables are going to appear significant when in reality they are not. So and also we added the accumulated amount of rainfall in the growing season and the number of days with soil temperatures below freezing. So to do that, we obtain daily data of temperature and rainfall from a dense network of weather stations in Nebraska, and in some cases we use on-site rain gauges. And the data for those fields were estimated by spatial interpolation. And this is something great in the United States, that there is a very nice, in most states, network of stations, and it's easy to access to those data and calculate by statistical methods or interpolation method specific weather data at certain places. So in this case, we defined that all the stations located within a 25-kilometer radius were used for the estimation of data. So as preliminary diagnostic, we observed the distribution of PF, the variance and mean relationship, and the correlation of the predictors. And what is the simplest? I'm measuring population densities, and what is the simplest way to deal with it? To use the percentage, I measure how much I have at the beginning, how much I have at the end, and I get a percentage. Why to deal with all this model? And this is unfortunately what has been done with traditional methods, using or trying to force a variable that is of a certain type into something that is not. And as the saying says, we can run, but we cannot hide. So in this case, final population, your number of eggs, is a discrete variable. We have 1,000 eggs, 2,000 eggs. We don't have 10.5 eggs, so it's discrete. So why I'm going to model this using the percent decline, that is a continuous variable, when this variable is naturally a discrete variable. So we model the final population as a response. So correlation used was as an exploratory analysis to see what variables may have some potential predicting capability. In this case, pH, the initial populations, texture index, and the number of soil days with temperature below freezing. So in the second step of the model, at this point we already defined, we are using a regression model. And you are gonna, just take this, just go with, by faith. But to come up to this, deciding this model was a process of diagnostics and estimation and specifications. Normally, when counts, which are very common, we measure in plant pathology, we measure number of lesions. In nematodes, it's all numbers. So counts are a discrete variable. And normally when counts are going to be modeled, we start with a basic model, which is a Poisson model. And then with a set of diagnostics, we come to a negative binomial, which has like a pedigree of 20 different variations of the negative binomial. And the most common is the NB2 parameterization. And so we use that type of parameterization. So the data set was randomly split into two sets. One set called the training data set to build the model in that data set, and a training data set to validate the model. So with those 8 variables, we feed a model with all of them. So and the second step is to, as we see here, there are some variables that appear significant, some marginal and significant, and some are not. But in the process of modeling, it's important to make sure that we are not missing out on some variables. I can say, okay, here these variables are significant, I'm discarding the other ones, I'm done. Well, there are some methods to make sure that the functional form of a variable is correctly put into a model. What about if accumulation rainfall is not linear, and I'm putting this in a linear form? How about if it's quadratic? So we use a method called partial regression plots. This is a scatter plot of residuals, conditional residuals. It was done for every variable. And I will not get into the details of that, but according to this assessment, all the variables appear okay on, appear to have a linear relationship rather than a quadratic or cubic. So a second model was fed with all these variables, which included the linear, quadratic, and interactions. And using automated backward elimination, we came up with a final model. So at this point, we are excited and say, okay, it looks like we have a model. This looks kind of complex, to interpret, and we have a relatively small data set. So let's evaluate. Let's see how this model looks good or not. So most common, evaluation of statistical model is a residual analysis. We examine that there are no specific patterns here, but more importantly, here is some observations that look out of look unfitted by the model. So we hear all the time also talking about outliers. We say, okay, there is an outlier here. But more importantly than an observation being an outlier is to make sure that that observation has some influence in the estimation of the regression coefficients of a model. So in this case, an observation 24, continued to pop up in all the assessments. I show two graphs here, but we use different methods to assess influential observation. So this table, I just want to call your attention on the last column. So there are some coefficients that after removing this variable from the model, this observation, not this variable, because the observation itself contains all the variables. Some regression coefficients, they were changed up to 100%. So without this assessment, we could have gone with a model that appeared good when in reality, it wasn't. So our final model is a model that contains pH and the initial populations as predictors. And all the assessments for that model show that this model was okay. So the model was validated into the data, a validation data set, and also into a training data set, and show high predicting capability. But after all this math, all this collecting the sample, how can I apply that? How can a grower, how can a producer, or how can I translate this information in a practical way for use to growers? So this constitutes the basic for risk analysis. For example, if a grower has a field with a population of 5,000 based on our model, and this is only in Nebraska because my population of inference is Nebraska, and so I expect the populations by the after annual corn rotation to be 1693. So a decrease or mortality of 3,300 is there. I put those in gray on purpose. So compared to a field that has a soil pH of 7, and the final populations would be of 2,500, around 2,500. So, or the results of this study show that pH and the initial populations are major determinants of that reduction after corn rotation. And applying the model specifically to that is that for any 1 unit increase in pH, the final population is expected to increase by 53.4%. And this is considering the fact that pH is, we say 1 unit, but in reality it's a logarithmic unit. So if I have a pH of 4, a pH of 5 is 10 times greater than a pH of 4. pH of 6 is 100 times compared to 4. So it's in a logarithmic scale. So for every 10% increase in population, in the initial population, the final population is expected to increase by 8.3%, holding the pH at its mean. So with this, I move to the second study that has to do with resistant varieties. And resistant varieties are an effective strategy to manage SCM. So every year in the United States, hundreds of varieties all over the major soybean growing areas are assessed for agronomic performance and response to SCM. The methodology is also simple and standard. A grid of neighboring plots, 4 rows, from 15 to 25 feet long. But only the 2 center rows of each plot are assessed. And yield is also measured only from the 2 center rows. In each plot, arbitrarily 10 to 12 soil cores are collected. And we ask this question. We assume when we are sampling those plots, we don't think about the spatial arrangement of the sampling entities within the plot. But we can ask, how about if there is a pocket of an aggregation of the nematode in the plot? How would that interfere with my assessment of a resistant or susceptible variety? How about if I have aggregation only on 1 row? what does it mean statistically? When we are sampling a plot, we unconsciously assume that at every soil probing we are collecting nematode or sampling units. So that statistically means that we are assuming that the population is random or uniform, and at every soil probing I will collect a propagule, so in this case cyst. These thoughts are not new, and the spatial variability has been recognized as an important component in sampling, but in the context of resistant varieties, it remains unclear how these entities arranged among each other is unclear. So our objective was to characterize the intraplot and interplot variability in experimental settings used to evaluate varieties in Nebraska. So simply put, or the goal of this study was to test the hypothesis of non-randomness. So field plots were established in 3 sites in Nebraska in 2012. Each experimental site consisted of a matrix of 6 by 6 plots, and each plot 10 feet wide, 15 feet long. And the sampling was conducted in intensive mapping. So first was a composite sampling. A segment of the plot was delimited, and at every 3.3 feet, a soil probe was collected, and those soil cores were put into a composite sample. And immediately, a single core sampling was conducted and attempted to be at least from 1 to 3 or 4 inches away from the initial. But in this case, for the single core sampling, every single core constituted an individual sample. And this is very difficult, is laboring to do in practice, because to avoid contamination, one soil core is collected, and the soil probe needs to be washed and rinsed, and then take the second, wash and rinse, and collecting above 1,000 soil cores is quite a bit of work. So for the intraplot, we use a distribution theory, beta binomial distribution. Again, I'm not going to get into the detail why this distribution is, why we pick the methodology. But something important to keep in mind is that when assuming that the methodology was picked correctly, this methodology gives you a value of the mean proportion, variance, and aggregation. So the interplot was studied using autocorrelation. So in each experimental area, each block, in each block, 2 transects in those 2 center rows were formed. So there were 12 transects per experimental area. In the 3 location, there was a location with 47 eggs per 100 cubic centimeter of soil, 406 and around 2,000, which we regarded as high. So the beta binomial distribution gives me 3 values. In this case, is the mean proportion, which is a value of the probability of in this case, of a soil probe containing sampling units, in this case cyst. So in the low mean density, the probability of going and sample a plot is 41%. In a moderate population density, the probability is 81, and in a high population, the probability was 94. And this is important because if I'm selecting a variety trial and I have a population that is low, maybe it's not worth it to establish my trial right there, based on certain, and this applies, of course, the level of inference or the scope of the inference for this is applied only for the areas of Nebraska. But this is the information that can be obtained from this type of analysis. So the beta binomial also indicates that nematode, the soybean cyst nematode aggregated in all this plot, intra and interplot. Even when there is a very low population density, aggregation already exists. So this graph just represents the variability. You know, these are the two rows of the same block. You can tell that in a row, a sample can be collected on this side, have no nematode, and 2 feet away, this sample have 5,000 nematodes. And the other shows the average range of dispersion. So according to this analysis, the average range of dispersion of the soybean cyst nematodes is of 9 meters. And in the standard for the plot size is 4 or 5 meters. So that's something that hasn't been considered, but the analysis says that this has some relevancy for obtaining important information about the assessments. The maximum distance was 15. So here, what this graph indicates is that population densities of SCNs are especially correlated up to 2 plots away, you know, at the plot, at the standard plot size. So in practice, that would mean for this particular site, it will mean that if I really, because yield in the end is assessed with a common analysis of variance. So analysis of variance, this is a big assumption, and this is strong in the statistical methods. We need to get a very good approximation, which is the purpose, and also make sure that assumptions are made. So varieties are compared for yield with analysis of variance, which it assumes independence of observations. So which, you know, this shows the opposite. Even when we have very small concentrations, population densities, there is some spatial dependence between plots. So rigorously speaking, this would mean that we have to skip every one plot to make sure that we are getting independent observations. So at the intraplot scale, at the interplot scale, the population densities occur in an aggregated pattern, and this also prompts consideration of this pattern to obtain better assessments of varieties. So with this, I finish my talk. Talking about my research, and I just want to briefly share with you what are my goals and my extension plans at this position at Uvalde. First, these are the goals that I consider attainable for this position, and my goal for research is to become a recognized authority on epidemiology and management of major diseases of agronomic and horticultural crops in the winter garden region and South Texas. And for the extension, my goal is to become a highly valued resource for growers, county agents, and stakeholders on disease management. How? Through innovative, applied research and collaborative research. Through successful project funding and effective communication and transfer of information to clientele. So this is the strategy, but now what are the tactics? What am I going to do? So my first goal is to familiarize myself very well with the extension system that is already in place, and with other extension programs that in the center and in all the 14, 15 centers of Texas. Liaise with extension and research faculty from the Uvalde Center and from other centers, with county agents, and get to know my clientele, urban clientele, growers, stakeholders, commodity groups. And the purpose is to visit with them and identify what are the needs, what is priority, what are the most important needs, and identify that to set up the right hypothesis and the right questions to try to answer those questions. So the focus of this position is disease management. In disease management, integration of biology, ecology, and epidemiology is fundamental. Just in the biology part, pathogen identification and disease diagnostic is big, is crucial, first step. So I see here a lot of opportunities, and I will work with Dr. Kevin Ong on disease diagnosis. And I'll bring my experience in molecular diagnosis of fungal pathogens to my skill set for this position. So, ecology and epidemiology, even in very simple experiments like a fungicide trial, before we compare a fungicide, before we compare an epidemic, we need to measure things. We need to monitor the epidemic. And that means to sample in the proper way and to assess disease in the proper way. And I have some experience with a spatial pattern, and spatial progress and temporal analysis of epidemics, and I will bring this to also as one of my skill sets for this position. Some type of studies that have immediate application. Fungicide trials, evaluation of varieties, fertilization, tillage and cultivation practice. And considering that water is an issue in that region, I'll incorporate studies to assess the effect of water, this irrigation with disease development. Some, leaf wetness duration and hydric stress, how hydric stress influence disease development. Sources to ensure that I'm going to have a successful funding, commodity groups like the Pecan Growers Association, the Vegetable Growers Association, and the Peanut Growers Board, Producers Board, and Water Developmental Board, competitive grants of IPM programs, the USDA NIFA IRS, and from industry. Just working for a small private company. I talked with Dr. Woodward this morning and say that companies are all the time willing to participate with universities, 100% of the time. So industry is another venue, so I will make sure to build up partnership with industry. So in summary, how this is, I will establish research and extension collaborations regionally and nationally, participate in multi-state committees, establish partnership with private industry, as I said earlier, ensure steady funding through several agencies I mentioned before, and present my research and extension at state, regional, and national conferences. And of course, publish my research in referee journals and my station information in state, national, and trade publications. In this case, the Plant Management Network, the AgriLife fact sheets, and of course, mentor students, national and international. So, with this, I finish the presentation. I thank you for your attention, and I thank also the people who contribute to my research, my PhD research. So.
We do have time for questions.
With the aggregation that you saw with the, how many reps do you need to do to get the resistant versus susceptible soybeans?
So the question is that with the aggregation observed, how many replications do we need to detect susceptibility or resistance? These are very important and practical questions, and the truth is that I don't know. But I don't know. But this study with probably with more data, it will be because one limitation is only three locations. But with more data, I'm pretty sure that it will give us a better sense of the number of reps that would be needed to detect differences. And this is important because in practice we are used to evaluate fungicide, insecticides, or 4 reps is the standard. But when we have a variable that the variation is very small, I need a lot of replications to be able to capture that variability. In this case, populations are all over, So maybe we may think, okay, I don't need a lot of replications to detect, to pick up that that differences. But the variation is spatial, so it makes the subject a little bit more complex.
So Oscar, there's been time now, I assume, for the soybean grower breeders to digest this kind of information a little bit. Do you sense that they're willing to change the way they set up their plots in light of this kind of data?
So the question is, if based on this data, if growers would be willing to change, or researcher or breeders
yeah.
to do, also a good question, and I discussed this subject with a plant breeder, with a student, a colleague from University of Nebraska, and we talk about this, and but this topic was studied a lot by a Greek person, and in 1960s, and then it was abandoned. So to a point, it has been neglected. So I don't know how willing they will be, but so I'm sure that more research is what will convince them. So that's our job is to bring that to them and show them, okay, here are the data. Are we willing to increase the number of replication? Because it also has to do with cost and sampling. But research wise, is it worth it to increase the number of replications?
Well, so is it working though? I mean, is the present method of screening for resistance identifying things that are working, or growers saying, okay, here's something that's supposed to be resistant, we plant it, we're seeing yield losses?
Yeah, also very important question, and from my perspective, is that the spatial component in the context of variety trial is unfortunately ignored. So no much attention is paid to this. So how much is that having an impact in those assessments? From what I see is that there is a huge impact, and so this information is to be taken into account, especially to decide, in a variety trial, to decide if this variety is indeed resistant to the soybean cyst nematode. Because some of the problems is that one grower says, okay, this variety at this site is resistant. Next year, the variety doesn't show the same type of resistant response as it did last year. And of course, it has to do with environmental, soil, and all that. But I believe that there is some factor, is related to, some of that is related to those inaccurate assessments. And with the soybean cyst nematode, also what complicates the issue is that it has HE types, which is equivalent to what we call physiological races. So one nematologist from Iowa State, he jokes and he says that you are in a field and you sample here and find an HE type, and then 5 meters away you find another HE type. So that variation is very high.
When you developed your model, you indicated that pH was one of the factors that you used in validation of the model. How much variability is there in pH within the data set that you have, and how do management tactics that are incorporated in a corn rotation influence soil pH compared to the soybean aspect of that rotation?
So the question is, if in this data set, what is the range of pH, and what other factors affect that pH?
In those two cropping systems.
In soybean and in corn?
Yes.
Okay. First, the range goes from 5.5 up to 8, the pH range. And how other factors affect pH? Frankly, I don't know. And in soybean, there have been some studies on texture, relationship of texture with soil pH. But my guess is that nematodes are essentially aquatic animals, and they inhabit water films that surround plant tissue or soil particles. So, and this is a guess again, is that is moisture what may have the highest relationship with soil pH, and probably the concentration of other of salts or cations, free cations like zinc, manganese in the soil.
In developing that model system, you kind of eliminated all the variables down to the initial population and pH. And you did mention too that that was from Nebraska. Yet I think Texas, we're a big state, and Nebraska is too. And there are a lot of different types of soil involved in other things. Now, obviously, with that, I guess, tree locations that you use, or I don't remember how many.
For the fields was 79 locations.
You managed to get a really nice equation down to 2. But how far do you think that will fit in terms of range of fitness of that? Do you think it's going to be a global thing? You think it's something that further research must be done in different areas, and if so, what needs to be looked at and how to simplify it instead of having to do it all over?
Okay. So the question is, if I understood it right, if this model is applicable to it can be applicable in other regions, or if everything has to be redone to ensure that actually these two variables are the driving forces for the reduction. And this is, we sample Nebraska, and so our population of inference is Nebraska. So we cannot extend these results to other regions. So it sounds sad, but a study like if we wanted to try something in Texas, then it has to data for Texas would need to be collected. So the model, in other words, the model will not be applicable to other regions, but to Nebraska only.
So I was curious about so that pH effect seems very strong.
Yeah.
Right. So I wonder if clearly you're done, but if there might be further. Number one, I wondered what direction is the relationship in pH. So nematodes do less well at lower pHs or higher pHs. And then the other question I had was there any plan to test the model with some kind of manipulation of pH?
That's a very important question. What is actually the effect of pH? In this case, we are talking about a year where the soybean cyst nematode doesn't have a host. So it's a non-host. So the situation changes a little bit when there is a host, because the nematode, first of all, the nematode is inside the host and, causing infection. But in corn, there may be cysts that break open and juveniles are released. So the nematode is exposed to an inhospitable environment. Perhaps soil pH is inducing hatching. It could be a factor during that non-host rotation inducing hatching. And so because the nematode, when it's in the cyst, is protected, but once the juvenile is exposed, it's vulnerable. It becomes vulnerable. So if that pH is causing some hatching, then it's likely that that mortality observed in rotation would be because of that effect. But as far as I know, there isn't any study like in vitro, and it's very simple but very practical to determine how soil pH. What is the effect of soil pH in hatching or survival, even survival? Putting juveniles in small petri plates and make sure what is and with temperature, probably mix it with combine that with temperature and see how those juveniles can remain alive based on the acidity of alkali.
Is this research at a place where it has a translational value to the grower? What with respect to you're finding about pH?
Yeah, and the,
Are we being too simplistic about what we could do with the data?
I believe that the model is applicable, and that indeed, and this confirms, some previous reports that pH is a factor that is related to the soybean cyst nematode. So I believe that it has applicability, and based on the validation, it looks like the applicability it has high applicability. So and that what is the use? How growers can use it? And I show that table where if a grower has a population of 5,000, say for example, speaking in practical terms, okay, I'm a grower, I have a population of 10,000 nematodes per 100 cc of soil. I have pH of 6. So according to the model, I expect more mortality, less mortality, right? So if I can tell a grower, okay, if you're testing for pH, we have a pH of such 6, 7, and your population is 10,000, so you expect a decrease of this much. So maybe I suggest for you, you have a high pH, very high risk of getting a lower population decrease with corn rotation. I would recommend to go to 2 years, but theory is easy, but practice is a different subject. We cannot change. In Nebraska, growers are very willing to intersperse soybean, for more than 2 years. So it's soybean corn, but that would be one of the. That probably increase the rotation, or do pH correction, because pH not only interferes with nematode but also with nutrition. Having a pH close to neutral is better in terms of plant nutrition.
So it seems like you very simplistically, you count total number of cysts at the initial population and then at the final population. And when you're doing resistant or tolerance trials, to me when I see resistant or tolerance to pathogen, especially something like a cyst nematode that has to get to the plant root, has to invade the plant root, has to put in its effectors, has to establish the relationship. It seems like resistance could occur at a number of different steps, or tolerance could occur at a number of different steps. Is there any way you can use this to start identifying traits and varieties that you can almost start compiling together to give an overall resistance? Let's say one has less attraction to nematodes, maybe the eggs don't germinate as well, maybe the nematode juveniles don't migrate towards the root as well because of exudate changes in those plants. Maybe another variety has less ability for the nematode to actually puncture and form the initial feeding relationship in the vascular tissue. It seems if you start identifying varieties with a number of those different characteristics, you can have much increased resistance to the nematode infection.
Yeah.
Can you use any of this to help you identify at what stage you're actually getting increased tolerance for nematodes?
That's an excellent question. And for soybean cyst nematode, there is a system of race identification. It's called HG typing, like H standing for heterodera and G for glycines. So in the United States, the problem with resistant varieties is that most of the varieties, 90% of the varieties grown in the United States come from a one resistant source. So in the 60s, a lot of germoplasm was imported from China to study resistance for soybean cyst nematode. And there are in the U.S. germoplasm bank 7 planting productions that are used in the U.S. But only one planting out of those 7, only one planting production is the predominant. For most of the resistant varieties come from that plant production for other reasons, agronomic features and stuff. So there is a very neat HG typing. So in the greenhouse, populations are tested against a standard susceptible. And probably, I don't know if Dr. Don has done or has seen that, but it's neat how and they determine that a variety that exhibit, they measure a female reproduction index. So if a variety, and this is set as a standard, if a variety exhibit reproduction of 10% or less, the variety is considered resistant. But if the variety has, from 10 to 30, moderate resistant, and then from down to 100%, if the variety is susceptible. So that's based on the H typing. And I don't know if I think I missed something.
No, actually, that was a good response.
But
It's not an easy question.
Go ahead.
one last thing is with nematodes is that in nematodes it's important to distinguish between total resistance and partial resistance, because it has been demonstrated that some varieties that show partial resistance versus total resistance. In a partial resistance, populations continue to increase after certain periods. Say, a total resistance, at 90 days, the populations reach a maximum. But with partial resistance, at 120 days, the population continue to increase. So it's important for nematodes in general, to distinguish between those two, partial or total.
Okay, I think with that, we'll go ahead and wrap up. Thanks again, Oscar.
Thank you very much.